We hope that it will help you understand what this technology is and what it can and cannot tell you. If you are deciding whether to have whole genome sequencing done, we hope this video will help you make your decision and help you decide what types of personal results, if any, you would like to receive from it. So, let's start at the beginning. In each of the trillions of cells that make up your body is your unique set of DNA. DNA carries all the information your body needs to function and makes you who you are. For example, how tall you are and what your hair color is. DNA is packaged up in 46 structures called chromosomes. Your complete set of DNA is called your genome. DNA is made out of 4 chemical bases called A, G, T, and C. These chemical bases, or letters, are the building blocks of DNA. The human genome is made up of about 3 billion pairs of these letters. When you stretch it out, it looks a little like this. The exact order of these letters is called your genome's sequence. Genes are specific important parts of this long length of letters. Humans have about 20,000 genes. Genes are important because they contain the instructions cells need to make proteins. Proteins do most of the work in your body, building and maintaining things like your muscles and organs, such as your heart. A change in a gene could lead to a change in a protein, and this could affect your body. For example, it could affect how your heart functions. About 99% of the letters in our DNA sequence are actually the same in all people across the globe. The 1% that varies between you and your neighbor is important, though. Although it seems small, this difference influences lots of things, like what you look like and your risk of disease, heart disease, for example. Your genome is what makes you human, but your unique set of DNA variants are what make you, you. Whole genome sequencing is a technology that enables scientists to literally read the exact order or sequence of all the letters that make up your complete set of DNA, your genome. In order to do this, a blood or saliva sample is taken. The sample is sent off to a laboratory where a machine breaks up your DNA and reads the letters like a code. Researchers then compare your DNA sequence to a standardized reference sequence. They identify the variants or differences between the 2 sets of letters. They then interpret the code. The process of whole genome sequencing can lead to many different types of personal information being identified. It may reveal DNA variants or differences in your genome, your complete set of DNA, that are associated with physical traits, such as height, eye color, and skin color. Some variants in your genome can give clues to where your ancestors lived. For example, some variants are found most frequently in people whose ancestors lived in Asia. If you have these variants, then it's likely that you have distant ancestors who came from that part of the world. You may also find out some information about your risk of developing common diseases, which are caused by multiple genetic, environmental, and lifestyle factors. For example, you may have DNA variants that mean you have an increased risk of a disease like type 2 diabetes. Remember, though, that type 2 diabetes is also influenced by what you eat and how much you exercise. There is also the possibility of getting personal genetic information about your risk of developing a psychiatric disorder, like schizophrenia or Alzheimer's disease. Whole genome sequencing can also provide information about how you might respond to medications. Variants in your genome can influence whether or not a specific medication will work for you, as well as whether or not you might experience unpleasant side effects from that medication. This is called pharmacogenomics. It may also reveal information about genetic disorders. These are rare diseases caused by abnormal variants or mutations in a person's DNA. One example of a disease caused by a mutation in this way is Huntington's disease. In most cases, if you have one or more of these variants, you will already have the disease, or will definitely develop it in the future. It could also reveal that you are a genetic carrier. This means that although you may be healthy, you have one copy of a DNA mutation in one of your genes that is disease-causing in people who have two copies. If your partner is also a carrier for the same mutation, then your children could be at increased risk of the disease. Sickle cell disease is inherited in this way. Finally, tens of thousands of other DNA variants will be identified. Although these all contribute to make you the unique person you are, scientists are currently unsure what role these variants play, if any, in health and disease. DNA variants in this category are sometimes called variants of unknown significance. Making your decision. If you are deciding whether to get whole genome sequencing done, it is important to know what types of results may be available to you. You will then need to weigh up the pros and cons and decide whether you would like to know all, some, or none of these results. The main potential benefit of having whole genome sequencing done is that DNA variants that have meaning for you personally may be identified, and you may have the option of getting these results. This might help direct you towards the right prevention, monitoring, or treatment for you. In addition, you may have the option of contributing your DNA anonymously to genomics research. By doing this, you could help make a big difference to what is known about the causes of disease. The main risks involve the possible social or emotional consequences of the information. There may be a loss of privacy, although the researchers will take precautions to prevent this. In some cases, a DNA mutation may be identified that indicates you will definitely get a severe disease that has no prevention or cure, such as Huntington's disease. In other cases, even if a DNA mutation that is associated with a rare genetic disorder is identified, how severe your symptoms will be may be unclear. It is also possible that you will be identified as having an increased risk of a disease like Alzheimer's disease, but not know for certain whether you will necessarily get it or not. The results could also have implications for your family members. All of these outcomes could lead you to feel a variety of emotions. It is also important to be aware of the limitations of whole genome sequencing at the present time. In general, knowing the complete DNA sequence of your genome does not, on its own, provide useful clinical information. The genomic data must be interpreted. This interpretation may provide some information about your health, but it also may not. This is because the science is still in its infancy. The first human genome sequence was only completed in the year 2003. Although the technology is improving each year, there is still much to be learned. In addition, interpreting a person's genome sequence is very complex, and scientists are still working out things like how to use this information to estimate a person's disease risk. So, whole genome sequencing may or may not have direct benefits to you. If you are considering getting whole genome sequencing done and receiving your results from it, it is important to remember that the decision is a personal and complex one. It is also completely voluntary and only done with written consent. If you do decide to get your results, these will be completely confidential, and it will be up to you to decide whether and to whom you wish to disclose them. So there you have it. Whole genome sequencing in a nutshell. We hope that you found this video informative and helpful if you are trying to make up your mind about getting your genome sequenced.
Hello, I'm Eric Chow. I'm an assistant professor at the University of California, San Francisco, and I'm going to talk to you today about next-generation sequencing. The outline of our talk today will cover traditional sequencing first, and then we'll spend most of our time talking about Illumina sequencing by synthesis, which most next-gen sequencing is being performed on today. At the end of the talk, I'll also touch upon two other competing platforms from Oxford Nanopore and Pacific Bioscience. So the Human Genome Project really spurred the development of cheaper sequencing. This was a 20-year effort that cost $3 billion that was completed in the year 2001. And this lowered the cost of sequencing a human genome to about $100 million for a single genome. And this used traditional Sanger sequencing. Before you go into Sanger sequencing, I want to tell you a little bit about DNA, just to bring everybody up to speed. So what I have depicted over here is a sequence of DNA. It's made of two different strands that run anti-parallel. So one strand runs in one direction, the other runs in the opposite direction. And your C's hybridize to G's, and your A's hybridize to T's. Sometimes DNA is depicted as an arrow shown here, just to simplify images. And we'll be using this quite a bit today. Now, DNA is a double-stranded molecule, but it can be denatured and separated into individual strands, and it can be renatured back together, or we can add on different sequences of DNA and hybridize those on, such as this red piece of DNA over here that's now bound to this bottom template strand. And DNA can be copied by polymerases, and these DNA polymerases, shown here in orange, can bind to short pieces of DNA that are hybridized onto a template strand, and they can polymerize DNA with building blocks to extend DNA into a newly synthesized strand. And these building blocks of DNAs are called deoxyribonucleotide triphosphates. And over here are the four different versions, your A's, C's, G's, and T's. And these all share several common aspects. First, there's a triphosphate group that allow the growing strand to attach onto the building block. They have a 3' hydroxyl group, which is used to add on additional building blocks of DNA. And they have 4 different bases attached to them. So with traditional Sanger sequencing, if you wanted to sequence a piece of DNA like this, you would have to first denature it and add on a primer so that DNA polymerase can bind on, as depicted here. And now, instead of adding just the traditional DNA bases, which would allow the polymerase to extend this DNA molecule, we actually use fluorescent terminators. And fluorescent terminators are very similar to the DNA building blocks depicted here. But there are a couple differences. First, you might notice that there are different colors, and so there's a different fluorescent group attached to each of the 4 bases. A yellow for A, a blue for G, a red for C, and a green for T. Additionally, the 3' hydroxyl group that's present on the building blocks to allow DNA to continue extending is now terminated or removed. This is why these are called fluorescent terminators. They have a fluorescent group and a terminator that prevents DNA polymerase from further extending the DNA strand. And so in a test tube, we actually have billions and billions of copies of this template. And we put in the normal DNA building blocks, and we spike in a low concentration of these fluorescent terminators. What results is you have a bunch of fragments of newly synthesized DNA that are all different sizes because they randomly incorporated a fluorescent terminator. And these molecules in the test tube are then put onto a DNA sequencer, which will separate these and then allow us to determine the sequence. What happens in the sequencer is these molecules are separated in size from largest to smallest. The smallest ones come out of the sequencer first and get detected, and then the next piece of DNA gets detected, and the next. And the trace that comes out of the DNA sequencer is a chromatogram down here at the bottom. And if you follow the color changes as each of the different pieces of DNA come out of the sequencer, you can build up your DNA sequence. These machines can run up to 384 samples at a time and generate about 700 bases of sequence for each of those samples. And you can sequence up to 1 million pieces of DNA or 1 million bases of DNA in a single day. Now, this might sound like a lot, but when you consider the human genome is actually 6 billion bases, and to sequence a human genome, you actually sample it many, many times, sometimes on an average of 7 times. This means that if you were to try to sequence a single human genome on one sequencing machine, it would take about 100 years. And so clearly, the Human Genome Project was completed in much under 100 years, and this was made possible by factories of these sequencing instruments. Over here is a single sequencing instrument in this huge factory where we have sometimes dozens or even hundreds of these machines running 24/7, 365 days a year. So the human genome was a really great undertaking, generated a lot of useful data, but it was only came from the genetic material of several people. And so we still don't understand most of what this genome does. We have the sequence, but again, it's only from a small subset. But to really understand the function of the genome, we need to sequence thousands to millions of different people to really sample the variety of genetic material present in the human population. And it's obvious we can't do this with traditional Sanger sequencing. So luckily, over the past 2 decades, the cost of sequencing has dropped dramatically. Over here is a chart showing you how much it costs to sequence 1 million bases of DNA. When the Human Genome Project completed in 2001, it cost almost $10,000. Today, we're approaching 1 cent to sequence 1 million bases of DNA, which represents a 1 million-fold drop in the cost of sequencing. And if you follow this chart, you can see that there are several inflection points in 2007, 2010, and 2015, where prices had some fairly steep drops. And these were largely driven by new sequencing systems introduced by Illumina, the dominant player in the next-gen sequencing market. On this slide are 3 different types of sequencers from Illumina. They actually have several more, but they really span the scale of sequencing that's being offered. On the left-hand side is the Illumina MiSeq instrument. In the middle is the HiSeq, and on the right is the NovaSeq, the newest high-output sequencer from Illumina. And if we compare the output of these machines and compare them with the Sanger sequencing platforms, we can see just how much more sequence we can generate from these instruments. If you just look at how many reads you can get from a single run, the MiSeq generates 30 million reads, the HiSeq generates 3 billion reads, the NovaSeq generates 13 billion reads, while the Sanger sequencing system generates about 400 reads. And so it's a huge difference. And if you just look at this in terms of how much sequence you can generate in a single day, you can generate up to 4 trillion bases on the NovaSeq in one single day, compared to 1 million bases on a traditional Sanger instrument. Illumina sequencing is the dominant player in the market. It's an imaging-based method and generates many, many reads, millions to billions of reads per run. And from each of these reads, we can generate 300 to 600 bases of sequence. It's really, really accurate. The error rate is roughly 1 in 1,000 bases. And with the new machines, we can actually sequence a human genome for $1,000 in less than 48 hours. And the sequencing on these Illumina platforms happens in flow cells. These are essentially microscope slides with channels on the inside. What I have shown here is a MiSeq flow cell next to a standard Eppendorf tube that's used for a lot of lab work. And these tubes are pretty small, about 1.5 inches in height. And on this MiSeq flow cell, we can generate about 1 to 30 million reads per run. Next up is the HiSeq flow cell. You can see it's quite a bit larger than the MiSeq flow cell, and this larger real estate allows us to generate more reads in a single run, approaching 3 billion reads. And lastly is the NovaSeq flow cell, which is even larger. And on this flow cell, we can generate 13 billion reads in a single run. So you can't just put DNA into these flow cells and get sequences out. You actually have to prepare your sample. And so there are several steps that occur, but basically what happens is you take your sample that you want to sequence, which is DNA. If you have something like RNA, you can convert it with enzymes into DNA. You take that DNA and then you have to add on these adapter sequences at the end. So in blue are primer binding sites that allow the sequencing reaction to occur. This is similar to Sanger sequencing, where we needed to have a primer bind for DNA polymerase to move along the template DNA. And then we also have these capture sequences in green and orange. And these allow your sequencing sample to be captured onto the flow cell for sequencing. Once you have your DNA sample prepared, it's a double-stranded molecule which we denature and put into the flow cell. And on the left-hand side, again, is a picture of the HiSeq flow cell. So there are 8 channels, and samples actually go into the glass slide. There are 8 different channels within there. And inside of the slide are a mixture of short DNA molecules in orange and green. And these orange and green molecules will bind to both ends of your sequencing library. So we denature our sample, flow those into the flow cell, and they can get captured by these primers or these short DNA molecules on the surface of the slide. And once that occurs, a DNA polymerase is added and the DNA building blocks introduced, and we copy that template. And so we have a newly synthesized strand that's now physically constrained to the bottom of the flow cell. We wash out the original template strand and then allow the newly synthesized strand to now bind onto the other DNA sequence present on the surface, in this case, the orange piece. And then we flow in some DNA polymerase and building blocks, and we get another strand formed. And we repeat this process many, many times, and in the end, we get about 1,000 copies within a cluster. And remember that these 1,000 molecules all came from the same original template strand, so they all have the same sequence. We denature the strands, and then we can selectively cleave off one of the oligos, or the primers, in this case, the green one. And we've washed those away, so now all the 1,000 molecules present are all the identical strand, because we removed the other strand. We flow in a sequencing primer, and now the clustering process is complete. And this is the instrument that performs clustering. So first thing we do is we take a brand new flow cell. Again, this is the size of a standard microscope slide, and then we put it onto the stage of the instrument. And the stage is a region of the instrument that can actually heat and cool to perform a lot of the enzymatic reactions. Next, we load a blue plate of the reagents that perform the clustering. And lastly, a strip tube with the 8 different samples that are going to be loaded onto that flow cell. Next, a manifold is mounted on top of the flow cell and connected to the CBOT. And in the back of the CBOT, or this clustering instrument, are a series of pumps that will now pull reagents from the blue reagent plate or from our sample strip tube and pass them through the flow cell. And this is how reagents and liquids are delivered. And at this point, the clustering procedure takes about 3 to 4 hours. And once that's done, we take that flow cell and move it on to the actual sequencer, in this case, a HiSeq instrument. And on the HiSeq instrument, we have a refrigerated compartment for all the sequencing reagents, a lot of enzymes in there that need to be kept cool during the 3 to 4 day sequencing run. Above the refrigerated section is a series of pumps that pull reagents from that refrigerated compartment and send them to the flow cell, which we will now load onto the stage. And so that is the flow cell from the CBOT that just completed clustering. This gets mounted onto a stage and locked in place. And then we can begin the sequencing run. And what that does is that will trigger the pumps to start flowing reagents from the refrigerated compartment into the flow cell. And behind the flow cell is actually a really powerful microscope that is used to actually sequence each of those molecules of DNA. And so remember that when we put the flow cell onto the sequencer, this is what it looks like. We have a lot of clusters that each have a sequencing primer bound to them. And the chemistry for sequencing is very similar to the Sanger sequencing terminators that were used. But the one difference is that these are reversible. And so they're reversible in 2 ways. So first, these can get incorporated into the clusters by a DNA polymerase. And those clusters will light up in 4 different colors depending on which base gets incorporated. A picture is taken, and after that picture is taken, we can actually remove these terminators and the fluorescent groups with some chemicals. And what results is we regenerate the 3' hydroxyl group. So now that cluster can have another round of bases added to it. And so this is what it looks like for a single molecule. On this end over here, we have our template strand bound to a sequencing primer. And this allows DNA polymerase to bind and then add a base. In this case, it's a yellow one. So once this gets added, a picture is taken. And then we add chemicals to remove the fluorescent group and the terminator so that DNA polymerase can add a second base. An image is taken, chemistry is performed to remove that base and to regenerate the 3' hydroxyl, and then we can add another base. And so over time, this cycle just repeats over and over again with multiple cycles of a base addition, imaging, and then chemistry to remove the blocks. And so if we take a look at this depiction of 5 different cycles, so each of these represents a different image taken. And if we follow the top left and the bottom right clusters and see how those colors changes over time, we can actually build the sequence. So the top cluster, the sequence is going to be A, G, C, C, T, because it goes from yellow, blue, red, red, green. And the bottom one is G, T, A, A, C. And so again, the power of the system is that you can sequence up to billions of sequences at the same time in parallel. And this is what gives these instruments their throughput. So some people ask me, why can't we increase the amount of bases that we get in a single read by just repeating the chemistry over and over again, do this 1,000, 2,000, 10,000 times? And the main reason why is that the enzymes and chemistries aren't perfect. So over here, I have a cluster, one single cluster made out of the same template molecules. But what happens is, because chemistry isn't perfect, some of the strands lag behind. So this one is a yellow instead of a green. And some clusters jump ahead. So this one is now a red instead of a green because it hopped ahead. And what happens is these errors aren't too bad, but over time and many, many cycles, over the course of 100, 200, 300 cycles, more and more strands start to lag and more start to jump ahead, so that your true signal starts to disappear and gets weaker and weaker. And this really limits Illumina sequencing to about 300 bases for each read. So with the Illumina sequencer, 4 different images are taken during each cycle, one for each of the 4 color bases. And so the images actually look like this. They're not very clear, they're not perfect circles, and they're very difficult to see by eye, to figure out where one cluster starts and another begins. You know, one example is here. Let's say in the first cycle we have 2 clusters that are both G. These just look like a single blob that's a single color, and this is really hard for the sequencer to pick out. But because we're sequencing many, many sequences at a time in parallel, chances are these aren't identical sequences. And if we go through another cycle, you know, chances are that they'll have a difference. So now this cluster on the right is an A, while the cluster on the left is still a G. But because of this, the sequencer can now compare these images and determine where cluster A has a very pure signal, where there's a mixture between cluster A and B, and where cluster B has a pure signal. And it'll decide to just take imaging information from just these clean areas where they have very pure signal. So again, this is 4-color chemistry from Illumina. This is what had been used for many, many years, and it was very clear and obvious, 1 color for each of the 4 bases. But there are issues with this. So the colors depicted here, you know, you can tell the colors are very, very different. However, if you actually look at the emission spectra, you can see there's significant overlap between the 4 colors. And